and welcome back to Newton Gate, where we make your dreams come true. Today, we will discuss about cell, the unit of life. So, what is cell? Cell is the basic unit of life. It's the fundamental structural and functional unit of all living organisms. Okay. So, first it has been seen and described by Anton van Leeuwenhoek. Then the nucleus, which is present inside the cell, is discovered by Robert Brown. So, this is the structure of the cell, and you can see here many membrane-bound cell organelles. This is the nucleus part, and this is the fluid matrix known as cytoplasm. So, basically, cell consists of three parts. Okay, a typical cell consists of three parts. The cell wall or cell membrane. Cell wall present in case of only plant cells. Cell membrane is present in both animal and plant cell. Okay. Then comes the next part, that is the cytoplasm, in which all the membrane bound organelles are suspended, and cytoplasm is the fluid matrix. Next is the nucleus, which is also termed as the brain of the cell. This part we will discuss in our next video. Today we will discuss about the cell. Okay, so why it is termed as the fundamental unit of life? Because human body consists of many organ systems. Okay. Now in every organ system consists of many organs, and every organ consists of different kinds of tissues, and a single tissue consists of millions of cells. So what is the connection here? This is the connection, that cell is the fundamental part of the human body. Okay. Here we have taken the example as human body, but you can take any other example also, like a plant or an animal. Okay, like this. Then we come to know about the cell theory. Cell theory, there are basically three postulates or the three main points of cell theory. So first two postulates have been given by Schleiden and Schwann together. They formulated the cell theory. However, they did not explain the last point, like all cells arise from pre-existing cells. So this last point has been concluded by the third scientist known as Virchow. So what are the three postulates? The cell is the smallest unit of structure of all living things, as we have discussed in our last slide, that it is the fundamental structure. Like if we break or if we study the whole organism, we will get cell as its fundamental unit. Now, cell is the unit of function of all living things. Why it is termed as the unit of function of all living things? Because it can perform all essential activities as single organism can, whatever he or she performs. Okay. And then comes to the next point, that all cells arise from pre-existing cells. Every cell arises from its mother cell. So it is a never-ending chain process. Okay, every cell arises from pre-existing cells. So these are the three postulates of cell theory. Next is the prokaryote and eukaryote part. So basically cells are of two kinds. One is prokaryote, another one is eukaryote. So we basically differentiate the cells on the basis of the presence of nucleus. In case of prokaryotic, there is no prominent nucleus. Okay. And in case of eukaryotic, there is presence of prominent nucleus. So there are other points also we will discuss. So first point is simple primitive cell type found in unicellular organisms like bacteria. So prokaryotic cell is the bacterial cell also. They are simple, they are primitive type, okay, and they are unicellular. Unicellular means they are composed of a single cell. Now what is the eukaryotic part? Like they evolved from prokaryotes. So the eukaryotes evolved from the prokaryotes part. They are larger and more complex. Okay. they are usually multicellular. Multicellular means they are composed of more than one cells. Okay, like plants, animals, etc. In case of prokaryotes, we see no well-defined membrane-bound nucleus. Nucleus is not that not that prominent. There is no membrane-bound. Okay, there is no nuclear membrane. But in case of eukaryotes, there is a nuclear membrane. So it contains membrane-bound nucleus. And then comes to the cell organelles. There are no membrane-bound cell organelles like Golgi body, mitochondria, etc. But in case of eukaryotes, there are membrane-bound organelles. Okay. So their DNA, prokaryotic DNA, is simple circular DNA. You can see here simple circular DNA in this nuclear portion, and then plasmids are also present. But in case of eukaryotes, their DNA are much more complicated. They are associated with histone proteins. Okay. And what is the reproduction part? They divide by binary fission. Prokaryotes divide by binary fission, which is a type of asexual reproduction. Okay. But in case of eukaryote, they divide by mitosis and meiosis. Okay. So this is the difference of division in case of prokaryotes and eukaryotes. Now there is no membrane receptors, but in case of eukaryotes we can see membrane receptors present. 70s ribosomes is also present, and 80s ribosomes is present in eukaryotes. They have single chromosome. Now eukaryotes have paired chromosomes. Okay. So basically these are the differences between a prokaryote and eukaryote. Prokaryotic cells. So bacteria, blue-green algae, mycoplasma. So these are all prokaryotic cells. Generally they are smaller and multiply more rapidly than the eukaryotic ones. And their sizes and shapes also vary. Okay. They have a cell wall surrounding cell membrane. Only we can see no cell wall surrounding cell membrane in case of mycoplasma. Now, nucleus is ill-defined in case of prokaryotic cells. Why it is ill-defined? Because it is naked, circular genomic DNA. There is no membrane, right? So it is ill-defined. It looks very it's not that prominent. Okay, it's an incipient nucleus. Some also have circular DNA outside genomic DNA called plasmid. There are some genomic DNA outside the circular DNA that is known as plasmid. Here which is known as plasmid, conferring certain unique phenotypic characters. Okay, they have got some phenotypic characters, that is antibiotic resistance. Phenotypic characters like the characters which are visible. Okay. There are various kinds of bacterial cells have been given, like Staphylococcus pneumoniae, that is diplococci, then rods shaped, spirals. Okay, these are the variety of shapes. Then comes to the next part, that is the cell envelope. Now what is it? Most prokaryotes have bacterial cells have a chemically complex cell envelope that acts as a single protective unit. So that is known as the cell envelope, that protects the bacterial cell or the prokaryotic cell, you can say. So what are the parts of a cell envelope? So it has got three parts: outermost glycocalyx, then cell wall and plasma membrane. Okay. So gram-positive and gram-negative bacterial cell wall. Okay. Here you can see cytoplasmic membrane, then cytoplasm. Cell wall is composed of peptidoglycan and lipoic acid. And glycocalyx, it is the glycocalyx part. And in case of this gram-negative, you can see the outer membrane. Cell wall is composed of only peptidoglycan. So this is the point of difference between the gram-negative and gram-positive bacteria, that in case of gram-positive, their cell wall is composed of two constituents, that is peptidoglycan and lipoic acid. But in case of cell wall, it is composed only of, in case of gram-negative bacteria, only cell wall is composed of peptidoglycan. There is no other component. Okay. So this is the next layer, that is the periplasm layer. So these are the two cell envelopes in two different kinds of bacteria. One is cell gram-positive and another one is gram-negative. Glycocalyx differs in composition and thickness. They have slime layer, that is this loose sheath, and the thick and tough called the capsule. Okay, so these are the two layers. What is the function of cell wall in prokaryotic cell? It's obvious. It determines the shape of cell and provides strong structural support to prevent bacterium from bursting or collapsing. It just prevents from bursting, okay, or collapsing. It provides or it gives a mechanical support. Now, what is plasma membrane in case of bacterial cell wall? Plasma membrane here acts as a selectively permeable in nature. We know that what is selectively permeable? Selectively permeable or semi-permeable membrane is that membrane which allows certain substances to pass through it. That is why it is termed as selectively or semi-permeable membrane, because it doesn't allow every substances to pass through it. Okay, it selects. This membrane is similar structurally to that of the eukaryotes. Next is the mesosome part. What is mesosome? It is special membranous structures which is formed by the extensions of the plasma membrane. There are certain extensions of the plasma membrane in the cell in the form of like vesicles, tubules and lamellae. So this structural extensions form this special membranous structure known as mesosomes. What is their function? What is the function of mesosome? They help in cell wall formation. Cell wall needs to get formed, so mesosome is the part that helps in cell wall formation. Now they also helps in DNA replication and distribution to daughter cells, secretion, respiration process, etc. They also increase surface area of plasma membrane and enzymatic content. Okay. So this is the diagrammatic structure of a bacterial mesosome. This is the cell wall and this is the plasma membrane. This part is the mesosome sac. Okay, this is the tubule. So this is the bacterial mesosome. Now bacterial cells has again been divided into two kinds. What is gram positive I have already discussed, and this one is gram negative. So what are the bases on which this two kinds of cell has been differentiated? Let's just see. Bacteria may be classified based on differences in cell envelopes and the manner in which they respond. Basically, bacterial cells respond to the staining procedure developed by the gram stain. So what is gram positive? No, the bacterial cells, those take up gram stain, those are known as gram positive. And what are termed as gram negative? The bacteria which do not take up gram stain, that is gram negative. So this is simple method. One is taking the stain, another one is not taking the stain. That is why gram positive, gram negative. Next is the ribosome part of prokaryotes. So ribosomes, as we know, that it is the site of protein synthesis. It is also associated with plasma membrane of prokaryotic cells. Now it has got two subunits, what we can say, and that is 50s and 30s subunit combining to form 70s subunit. Okay, so S stands here for Sedberg's coefficient, that is sedimentation coefficient, or you can say an indirect measure of density and size. Next is the polyribosomes, which is present in prokaryotes. This is what are polyribosomes? It is also termed as polysome also. Here you can see this is a single mRNA. So single mRNA, and here are ribosomes attached. Many ribosomes are attached to this single mRNA chain. So these ribosomes translate mRNA into proteins during protein synthesis. So this is termed as polyribosomes. When several ribosomes attach to a single mRNA structure, then it is termed as a polyribosomal unit. That helps ribosomes translate mRNA into proteins during protein synthesis. in prokaryotes reserve material stored in cytoplasm. Okay, they do not bound by membrane system and lie free in cytoplasm. They are also found in synophysin granules, glycogen granules, etc. Then gas vacuoles found in blue-green algae, purple photosynthetic bacteria and green photosynthetic bacteria. So these are the examples. So basically inclusion bodies are the bodies that reserve food materials. Okay, reserve stored material in the cytoplasm of prokaryotic cells. Now this is the structure of a eukaryotic cell. So eukaryotic cells consist of many membrane bound structure. Okay, it is a much more complicated structure compared to the prokaryotic cell. So all proteins, plants, animals and fungi consist of eukaryotic cells. They are compartmentalization of cytoplasm through presence of membrane bound organelles. Okay, then they have organized nucleus with nuclear envelope. Genetic material organized into chromosomes. So in eukaryotes, genetic material is very organized, but in case of prokaryotes, we haven't seen any organized genetic nucleus because the nucleus part is naked. Okay, so there is no membrane bound nucleus. Then variety of complex locomotor and cytoskeletal structures are also there. Then locomotor and cytoskeletal structures are also associated with eukaryotic cells. Now the next part is the plant cell and animal cell. Okay, the difference. Now what is the difference? Distinct cell wall has outer boundary and just within is cell membrane. In plant cell we can see a distinct boundary. Okay. Animal cell membrane is outermost limiting structure of cell. So cell wall is there, then cell membrane comes in case of plant cell. But in case of animal cell there is no cell wall. Only cell membrane is the outermost part. Plant cell consists of chloroplast with ribosomes. So chloroplast is the structure that is associated with photosynthesis. As the green plant capture sunlight with the help of this chloroplast. Okay, that helps the plant to produce their own food. But in case of animal cell, they do not have any chloroplast. Okay, so chloroplast absent. Now one large central vacuole is present. There is a large central vacuole, you can see here, is present. But in case of animal cells, there are many small vacuoles present. There is no large vacuole and they are very few in number. So in case of plant cell, there is no centrosome or centrioles present. What is the function of centriole or centrosome? They help in cell division part. So in plants, there is no centrosome. But in case of animal cells, we can find centrosome or centriole part. Now plant cell has a large and a fixed regular shape. Okay, but in case of animal cells, they do not have any fixed shape. Nucleus in case of plant cell lies on one side. So here you can see at the almost in the periphery of the plant cell. Okay, but in case of animal cell, nucleus is in the middle portion, the central portion. Okay, lies in the center. Few mitochondria are present in case of plant cell. But in case of animal cell, mitochondria are present in large numbers. Okay. Plastids are present in case of plant cell, but plastids are absent in case of animal cell. So we will discuss, this is all about cell, then prokaryotic cell, eukaryotic cell, differences between plant and animal cell. In our next video, we will discuss about the next part. So okay, please like, subscribe and share. Follow us on Facebook, Twitter and Instagram. For more details, visit our website www.newtongate.in or call us on the displayed number. Thank you.